Okay, now you can go ahead and get started, Hamid.
Thank you for attending this defense. Especially thank you to all the defense committee members. My presentation content is aligned with my thesis chapters. Each chapter is supported with 1 or 2 journal publications. Here is the content that is covered in this presentation. This is an overview image of my work, CMOS capacitive sensing platform for cellular and molecular monitoring. It includes design and implementation and also test of new hardware and software, where hardware itself consists of on-chip and off-chip electronics. On-chip or integrated circuit design part of the work includes capacitive electrodes and interface circuitry on a single CMOS chip. And also off-chip hardware, on the other hand, is an embedded system actually having a control unit and data acquisition to support the chip. And the software is a graphical user interface that allows for different user-configured experiments and real-time data visualization. Now, the question is, what is the application of CMOS capacitive sensors in life science? One major application is cell monitoring. The settling, attachment, proliferation, and growth of cells are important to biologists. In these stages, the integration of cells with the surface through multi-protein structures cause change in the capacitance. In this application, 2 types of capacitance, namely offset and target, exist. Offset capacitance has been reported in many publications to be in the range of picofarad and is usually due to parasitic capacitances, addition of solution, and remnants. On the other hand, target capacitance variation is in the range of sub-femto to a few femtofarad, according to the literature. The main cause of this capacitance change is the attachment of cells that are under investigation. A CMOS capacitive sensor in these applications must be wide enough and have sufficient resolution to measure both of these types of capacitances. Another application is molecular monitoring, such as in DNA detection. Typically, in these applications, the complementary strand of target DNA is immobilized on the sensor that is known as recognition element. Hybridization of the 2 complementary strands can change the capacitance as a target for measurement. Offset capacitance also exists in this one too, due to parasitic capacitances, as well as medium and remnants. The offset and target capacitances of different publications are shown in this slide. Based on these 2 major applications that we have, the main challenges in the design of CMOS capacitive sensors for life science applications are sub-femtofarad resolution, hundreds of femtofarad dynamic range, detection of variable offset capacitance, and creating an array of sensors for pixelated monitoring. In this thesis, we have addressed these challenges. Our solution to the first 2 challenges has been published in 2 journal papers. Let me start with the first one, published in IEEE Transactions on Biomedical Circuits and Systems. In this paper, we have proposed the circuit and system shown in this diagram. The on-chip interface circuit consists of an analog part followed by analog to digital converter blocks. The core of the analog circuit is a DC to DI conversion block that converts differential capacitance to differential current and is called charge-based capacitance measurement circuit, or CBCM, which is shown in this schematic. You can see an unknown capacitor, CS, and a reference capacitor, CR. CS and CR. Phi1 and Phi2 are non-overlapping clock pulses. By this circuit, the unknown capacitance can be measured by monitoring the 2 currents, IS and IR, using benchtop ammeters. The relationship between the average differential current and the differential capacitance is linear. The first effort to make an on-chip circuit with CBCM is the circuit shown in this schematic. This circuit mirrors the currents and integrates them in two different integrating capacitors, Cint plus and Cint minus. A differential amplifier is then used to create a single end output from the voltages created on the integrating capacitors. This circuit, however, yields limited dynamic range. To improve the dynamic range of the previous circuit, another current mirror composed of M9 and M10 has been proposed to added in this circuit. The magic is the added current mirror creates a differential current, and then the differential current gets integrated in a single integrating capacitor or Cint here. The result has shown increase in the dynamic range due to eliminating offset / common mode voltage. This circuit is named current differential CBCM. Current differential CBCM has been popular since then. So another group doubled its dynamic range by adding another set of current differential pairs to create a negative output voltage. The resulting differential output had twice the dynamic range. The dynamic range of these circuits were still in the range of a few to some tens of femtofarad. Here is our design proposed in the thesis. We've used an op-amp on a current differential CBCM and eliminated the integrating capacitor. This is a complete schematic of the proposed circuit that we have called enhanced CBCM or eCBCM in short. The circuit shown in gray is the current differential circuit without the integrating capacitor. The question is now, okay, now how does this proposed circuit increase the dynamic range? It does so by increasing the linearity for a wider range of input. The key point has been eliminating the integrating capacitor. Current mirror composed of N1 and N2, is there to copy the current of reference side I prime R to create a differential current I prime IDE minus I prime R in here. At the same time, the voltage of the integrating capacitor is the drain source voltage of N2. Any change in this voltage, therefore, directly introduces error in the linear functionality of the current mirror. The range of linear functionality of this current mirror will be improved by keeping the voltage of VD and VC equal. On the top left, we have shown simulation results for the non-enhanced current differential CBCM with an integrating capacitor. As can be seen, VD completely deviates from VC. On the bottom left, simulation results for the eCBCM circuit is shown. In the eCBCM, both VD and VC have the same waveform for a wide range of CIDE change as shown in this figure. Here, these equations represent the transfer function of the eCBCM circuit. With some considerations in the sizing of the devices, the pseudo-linear transfer function becomes a linear function where the average of the output current here is linearly proportional to the difference of the capacitors or the input differential capacitor. A comparison between the linearity of eCBCM and current differential CBCM is shown in the simulation results, which we have used real sizes from literature. eCBCM shows a significant improvement in the linearity. To create digital output from the analog differential output, or I-out, a current controlled oscillator, or CCO in short, has been used. The input of the oscillator is I-out of the eCBCM. The topology of the CCO has been adopted from literature. Then a counter serializer is used to generate a serial digital output by counting the number of pulses generated at the output of the CCO in a given time window, which is governed by Phi1 and Phi2 signals. Now that we have all the blocks of the system, the next question is the transfer function of the digital output over differential input capacitance. The digital output is the number of pulses, and according to this derived equation, the input has a linear relation with the input differential capacitance. Then we can calculate the equation for sensitivity. The sensitivity, we can derive the equation for sensitivity. The sensitivity of the entire system is calculated by differentiating the output over change in the input differential capacitance. This gives us the parameters that play a role in the sensitivity given in this equation. For your attention, the electric equivalent model of biological samples is slightly more complicated than a single capacitor. Therefore, the transfer function will be more complicated. This one has been investigated in detail in an empirical approach via simulations and experimental results. Here, the linearity of ECBCM transfer function has been investigated with post-layout simulation results. Results are shown for a large range of input differential capacitance for a given CR. High linearity is especially observed in about 400 femtofarad of the input change. The numbers are a squared correlation index in percentage or R squared %. We also want to see the linearity for a fixed CIDE, where the value of reference capacitor is swept. Another post-layout simulation is performed for this purpose. The linearity is seen for the full range as long as CIDE stays larger than CR. This is expected from the theory of operation of CBCM. Moving forward to characterizing the CCO, another post-layout simulation is shown here. The curve shows the number of pulses that the CCO creates in the 10 microsecond measurement window with respect to the average of the input current. A linear region is observed, then the number of pulses decrease in a nonlinear fashion and completely dissipates for larger values of input current. Now, since we know that the sample can have conductive properties, we have looked at the effect of resistance on the output waveforms of ECBCM. It can be seen that resistance can alter the waveform. For instance, part of the waveform in high conductivity solutions, such as in blue, transitions to the range of non-oscillating area. Another observation is that the waveforms expand when the conductivity is in the range of medium to high or medium, that is shown in the yellow curves here. Similarly, we have studied the change in sensitivity with respect to solution conductivity. Interestingly, it's seen that the sensitivity drops by increase in the conductivity of the solution. Now, I'd like to summarize the contributions of paper 1, which have been design, implementation, and fabrication of a new fully integrated circuit with an increased dynamic range and a sub-femtofarad resolution. Now, before sharing the experimental results of this chapter, I would like to present the second paper published in IEEE Transactions on Instrumentation and Measurement Journal, which is also included in chapter 2. Now, for this one, let's start with this diagram that illustrates the equivalent circuit model of the sample on chip. All the capacitances, including target and offset, and most likely resistance are shown. The total capacitance is equal to offset capacitance, C0, shown here, plus the target capacitance, or sigma C. The question based on this model is how can we measure the small change in the target capacitance if offset varies during the experiment due to remnants, change in the pH, evaporation of sample solutions, etc. There are a number of techniques to address this challenge. One of them is to equip the circuit with calibration, or more precisely, recalibration mechanism. For example, in this circuit of chip resistors, RP1 and RP2, have been used to manually adjust the amount of current in the reference side to compensate the effect of parasitic elements and mismatch. In addition, for further fine-tuning with the program, within the program, a current mirror with programmable gain has been implemented on the reference side using a number of switches connected to different CMOS devices. In this work, we have addressed this challenge by proposing a different approach. The image shows a bank of capacitors realizing a digitally programmable reference capacitor that has been replaced with the traditional fixed reference capacitor. Now, let's first discuss why continuous calibration seems important in life science applications. In this diagram, green particles demonstrate the target cells for which we want to measure the capacitance change. After a while, other large particles shown in orange can form or sediment that result in a large change in the offset capacitance. In general, various amounts of different particles can be sedimented during different measurement times. Generally, detection of a small capacitance when the offset changes significantly is impossible because your device will not have enough output range to sense that. Unless we have a mechanism to be able to capture all the changes in different ranges. The proposed technique relies on performing multiple measurements within all reference values and then some data analysis. Such a technique is visualized in this figure, where variation of output current as a function of reference capacitor has been plotted over time. Different offset values are then reflected in different times in the generated 3D plot. This is the basis for calibration-free measurement, since by this method, all possible offset values are scanned in a single measurement thread. Now, we are interested, based on the calibration-free technique, we are interested in possible patterns of digital output with respect to sweep and CR. As the effective conductivity was shown for ECBCM output patterns in time domain, here also the patterns become more versatile in the presence of solution conductivity. You can see post-layout simulation results showing 2 different patterns out of 4 possible categories, and highly conductive, this one, and in low conductivity solutions. And in this one, the other 2 patterns are shown for the low to medium conductivity and for medium to high conductivity. Now, let's look at the first fabricated chip consisting of 2 electrodes. This chip occupies 1 millimeter by 2 millimeter silicon area. The active sensing area is 0.22 millimeter by 0.3 millimeter. We have repeated the fabrication of this chip in 2 different runs to have passivated and non-passivated electrodes. This is our implemented off-chip electronics on PCB and the graphical user interface for the first chip. You can see the full system with the partially encapsulated chip. There are different options for the user to configure the test conditions, such as the type of test, the number of measurements, etc. This is the first electrical characterization results of the fabricated chip with no sample for both right and left electrodes. The patterns agree with the simulations for low conductivity solution, which in this case, there is no solution on the chip. Here, a 3D profile is obtained with a droplet of water. Different patterns can be seen as the droplet has been put on the chip until it has completely evaporated. This might be a 3D fingerprint for the evaporation of this water droplet. Another experiment, we could see the effect of salt, significant amount of salt, on the electrode. The after curves are for the case that salt has been completely dried. In this figure, we compare the simulation and experimental results using different samples. 3 different patterns can be seen for liquids that have different dielectric constants as well as conductivities. The patterns match with those expected for different solution conductivities. Real-time measurement results for the water droplet, when the recording starts before putting the droplet until it completely evaporates, is shown in this video. There is a baseline when there is no sample on the chip. This is what happens in real time after putting the sample. At the end, different waveforms can be distinguished in this experiment that are shown with different colors. These waveforms are baseline that has been captured before putting the sample, which is curve A. Other patterns are seen at the onset of putting of the droplet, curve B, contact of droplet with the electrodes, curve C, gradual evaporation and increase in the conductivity, D, onset of complete evaporation, E, and dryness with salt remnants in F. In a similar experiment with the 2 microliter evaporating droplet of propanol, this 3D profile has been generated. The gradual increase in capacitance as the droplet evaporates and the change in the pattern at around 130 seconds due to increase in the conductivity can be seen. This 3D profile is related to 2 microliter methanol droplet. This profile is different from the one we have for propanol due to different dielectric constant and conductivity of the two solutions. Another experiment is with tap water that shows a completely different pattern. These profiles might be considered as fingerprints of evaporation of these chemicals. What's important is that all phases of evaporation from baseline until the end, when it completely evaporates and the onset of evaporation also can be detected. The list of contributions include in the proposal of calibration-free measurement technique, and the experimental results summarizes this paper as well as chapter 2. Now, let's just start with the third paper that constitutes chapter 3 of the thesis. In this paper, we have addressed the challenge of array of capacitive sensors. In order to develop an array of electrodes, authors in this reference have repeated the entire core CBCM circuit, including its reference electrode for each pixel. The core CBCM is connected to the rest of the circuitry using 2 CMOS switches, shown here. In total, each pixel has 6 transistors and a reference capacitor. In another work, as shown here, multiple switches have been placed to multiplex the IDEs to a shared CBCM circuitry. Well, the result is footprint has been drastically decreased due to using a single shared CBCM. As you can see, in both scenarios, analog switches have been put somewhere in the path whose current is being measured. Therefore, parasitic capacitances and leakage of these switches can introduce unknown inaccuracy. To avoid this problem, we have proposed a circuitry where each pixel includes only half CBCM and there is no analog switch for the multiplexing. Instead, a digital multiplexing circuitry is used to multiplex the clock pulses to the active and non-active pixels, as shown in this schematic. These are the clock pulses generated by the digital multiplexing system. While active IDE receives Phi1 and Phi2 signals, non-active IDEs are all connected to ground. Now, the interface circuit is adapted from the eCBCM of the first chip by increasing the current gain between I2 and I out. As the post-layout simulation results show, when I' IDE waveform goes higher than IR, which happens in this curve here, I2 increases, and due to the high gain, I out jumps to a high current value. From chip 1 to chip 2, the resolution of bank of capacitors has also been increased from 7 bits to 10 bits. Here we see the linearity of the transfer function of the eCBCM of the array chip. The dynamic range can be obtained from the curve, which is around 100 femtofarad. For showing the sensitivity of the ECBCM, a 0.5 femtofarad step change of input has been simulated. The results show 0.59 microamp per femtofarad change in the average of current, which shows an increase of about 3 times compared to one. The current waveforms in time domain for a 1 femtofarad change in the input differential capacitance is shown on the left side. The red curve relates to a higher input, and its average is higher as expected. Looking at the post-layout simulation of the CCO, it can be seen that we have increased the oscillation range compared to the CCO in chip 1, while the topology has been kept the same. Combining the sensitivity of ECBCM and the oscillation frequency and increased sensitivity of about 7 pulses per femtofarad has been achieved. Another post-layout simulation has been performed to show the uniformity in different pixels in the multiplexing scheme. Different IDEs from different locations of the layout have been selected. Different amount of capacitances have been applied to them, and the average of output has been calculated and plotted. The curve shows insignificant amount of deviation among the pixels. That is an indicator of uniformity of the multiplexing structure without the analog switches among different pixels. This is the microscopic image of the fabricated chip. The second one, the same technology that we use for chip 1 is used in this one too. It has 256 electrodes in a 1.6 millimeter by 1.3 millimeter active sensing area. Half of the electrodes are passivated and half are non-passivated in a total of 2.9 millimeter by 2.5 millimeter chip. Dedicated hardware and software is designed and implemented for the second chip as well. Both hardware and software support multiplexing signals for channel selection. Software allows for full page scan mode as well as a single pixel measurement. It's the first electrical characterization results of the fabricated chip without any samples. Capacitance of the passivated chip is slightly higher as expected due to the coverage of the passivation layer. Among 256 electrodes, 2 of them are connected to the circuit via a transmission gate or switch. As can be observed, transmission gate adds more offset and slightly more nonlinearity. The first experiment with this chip is done by putting a 0.3 microliter water droplet manually on the chip using a micropipette. After full scan of the electrodes and subtraction with the baseline, a heat plot is generated. The heat plot and the droplet image show match in the location. This confirms the functionality of the proposed multiplexing scheme. The second droplet is a 0.5 microliter mix of 10% ethanol in pure water. The results confirm the location as well as the smaller numbers in the active pixels. This shows the sensitivity of the chip to the electric liquids, the dielectric of liquids, recalling that ethanol has a smaller dielectric property compared to water. The third droplet is a 0.5 microliter mix of 10% methanol in pure water. Apart from matching the location of the droplet, the numbers of active pixels are also meaningful for methanol water mix. More clearly, by focusing on the values of active pixels for the 3 mentioned liquids, the average and standard deviation is shown in this figure. It can be seen that the output of the chip increases, increasing the value of the electric of the liquid. In the list of contributions shown here, we summarize this paper in chapter 3. We've designed and implemented an array of 256 electrodes with a new multiplexing scheme and adapted the ECBCM and CCO circuit topology. Experimental results were shown, and they proved the functionality of the multiplexing, and we showed the sensitivity of the chip. Now we move forward to our 4th publication, which is the basis for chapter 4. In this IEEE Sensors Journal publication, we've reported our results for a new DNA detection paradigm that can be used in data storage devices based on DNA. Recently, DNA memories have gained attraction among tech giants such as Microsoft and Google. The main advantage is a significantly higher data storage capacity in the range of terabyte in a nanogram of DNA molecule. Among multiple steps that is required in this technology, physical storage and retrieval is a serious unsolved issue. Monitoring the amount of DNA during wet/dry cycles of storing and retrieving has been a bottleneck for device miniaturization. In this work, we have addressed the challenge of quantification of DNA in the dry phase without recognition element with our CMOS sensor chip 1. These equations pave the way toward understanding the existing offset capacitors and the total capacitance that exists when there is no sample on the electrode. Here we can see how the percentage of coverage of DNA, which is known as confluence, can change the total capacitance after deposition of dry DNA on the electrode. This is the experimental results. Using chip 1, we have obtained the 3D profile of aqueous sample with DNA, shown on the top left, and without DNA, shown on the top right. Microscopic images of the electrodes show DNA colonies remaining on the chip after evaporation of a 0.7 microliter droplet that contains DNA. In this image, we see capacitance readings which show different values at the onset of droplet evaporation here for droplets with different concentrations of DNA. These readings have been used to obtain the transfer function after repeating the experiment for 5 times for each concentration, 45 nanogram per microliter, 80, 117, 222, 287 nanogram per microliter. A linear transfer function between the change in capacitance and the concentration of DNA has been observed. In short, in chapter 4, we've shown the possibility of detecting DNA in the dry mode in the scale of nanograms with our CMOS capacitive sensor. Let's move forward and explain the 5th publication covered in chapter 5, which has been published in Bioengineering Journal and reports our experiment results for the use of CMOS capacitive sensors in monitoring oral cells. Recent studies show that gum diseases can be an indirect indicator of more complicated diseases such as diabetes, osteoporosis, and HIV. From another side, research has shown a link between oral polymorphonuclear neutrophils, or OPMNs in short, and gum diseases. The challenge is, unlike other cells, oral cells have very low culturability, even if they are in the culture medium. We've shown that these cells can be detected with the CMOS capacitive sensor. Here we see the test bench prepared for the real-time data acquisition using the GUI and capturing images before and after experiment by employing the high-resolution reflective camera. The equivalent model is illustrated for 3 distinct phases that exist in this experiment. The ionic sample solution has a high conductivity, and the proper time for measuring the cells is the onset of evaporation when there is a small amount of moisture that keeps the morphology of cells. Here, we see the images taken after evaporation of droplets. Images from A to E show the accumulation of cells after each time addition of 1 microliter sample. According to control hemocytometry results, there were 7 neutrophils and 13 epithelial cells in each 10 microliter sample. Different distributions of cells are seen on the right and left electrodes based on the hydrodynamic of evaporation. Image F is when the cells are washed from the surface. The output of the chip for each of the runs is demonstrated for right and left electrodes. A correlation between the amount of cells that covered the surface of electrodes after each run and the change in the capacitance is observed. In summary, experimental results were presented showing that the CMOS capacitive sensor chip can be used for measurement of the amount of oral cells attached to the electrode. This opens up a new avenue for measuring cells that cannot be cultured, such as oral cells. Despite all the improvements and achievements. Further steps are explained in this section that can help move toward a more advanced CMOS capacitive chip. One of these steps is to develop a microfluidic structure. A manual positioning of sample can introduce inaccuracy, for instance, in the volume of sample. A proper microfluidic device can help automate sample positioning as well as reaching high throughput applications where many different types of samples can be tested at the same time on the same chip. Another improvement step is to increase the spatial resolution of array. The crosstalk between the pixels has been a limiting factor in the minimum pitch that electrodes can be positioned. This problem is worth investigating in future works. In addition, in future designs, addition of a simple processing core with memory and PWM signal generator peripheral can be considered. This can make data acquisition and cross-platform data transmission much faster without the need for off-chip electronics. Furthermore, for some applications, including antigen antibody tests, additional recognition layer can be considered for adding selectivity to the chip for future applications using this chip. Here is a list of my journal publications. There are 6 publications in total. 4 papers have been published associated with the thesis. 1 paper from the thesis, for that one, a review has been submitted. We hope for acceptance of that one too. 3 conference papers associated with the thesis is listed here. I've also contributed as a co-author in 4 other journal papers with the lists that you can see here. A list of contributions of this thesis is listed here. In summary, 2 fully functional chips have been fabricated. 2 embedded boards have been developed for those chips. In addition, I have proposed new circuit blocks in each of the chips. Plus, we have done experimental results, and in addition, the possibility of using the CMOS capacitive sensor in 2 new applications has been demonstrated. At the end, I would like to highly acknowledge tremendous and enthusiastic support of Professor Ghafarzadeh in all aspects of this research. Also, I would like to thank all colleagues without whom doing this work was not possible. Especially thank you to thesis committee members and defense committee members, Professor Magrovsky, Professor Kassiri, Professor Moshiri, Professor Sanati Nejad, Professor Askari, and Professor Salahamish. These are some of the list of some of the references that we used in this presentation. And this is all for my presentation. Thank you.
Okay, thank you, Hamed. Thanks for keeping it within 40 minutes. That's great. I really appreciate that.
Thanks.
Without actually increasing the pace too much. So I really enjoyed your presentation. Let's get started with the questions. I'm going to ask well, let me actually first stop recording.